Hello everyone, my name is Rodolfo Probst. I am a graduate student at the University of Utah. First of all, I'd like to thank the organizers for this opportunity. And today I'm going to tell a little story about ant-plant symbiosis. As you're probably going to see through this meeting, natural selection can promote repeatable outcomes in evolution. And evolutionary determinism, or as I like to mention as the product of this conspiracy between adaptive radiation and selection, can be found in many systems. For example, in all the lizards, mandarin snails, ferns. I would argue that ant-plant symbiosis might be another example of that. With ants and plants having a tight evolutionary history, and we know that ants have been nesting in plants for at least 90 million years. Some of those associations between ants and plants are highly specific and involve myrmecophytes, plants that evolve with structural adaptations that provide ants with food and shelter. And most of our knowledge comes from the study of those conspicuous systems involving myrmecophytes. But I would argue that a whole universe of ant-plant symbiosis is waiting out there in nature. They might be comprised by inconspicuous partners. For example, the system I'm going to mention today, involving ants from the genus Myrmelachista and their hosts, plants from the family Lauraceae. But who is Myrmelachista? Myrmelachista is a lineage of tiny arboreal ants that can be found from Mexico to Patagonia. And in a recently published phylogeny, that genus was cover-assisted to brachymyrmex, with that clade being sister to the remaining formicides. What comes in handy is that Myrmelachista occupy what I like to refer as the full range of nesting habit. And what I mean with that, we can, based on natural history, you can roughly divide the genus into main groups. For the first group, we have species that are either dead stem nesters or live stem nesters, opportunistically. And so we have species nesting in plant cavities or dead stems. We also have species living in live tissues of plants, but they occupy a wide variety of plants and they don't have any specific association with them. And for the second group, the group of specialist or obligatory symbiotic species, we have species that can only be found nesting in a particular set of plants. And within that group, we have the famous devil's garden ant in North and South America, and those ants systematically poison saplings from the plants that are not the host, creating this monotypic patch of the host plant in the story of rainforests. And also within that group, we have a bunch of species across Central America that can be found nesting inside understory plants of the family Lauraceae. Now that I got introduced to Myrmelachista, I'm going to spend the rest of my talk mentioning the phylogenomic reconstruction I'm doing for the whole genus, how I'm using the topology to ask questions about evolution of nesting specialization by running ancestral state reconstruction analysis. I'm going to talk about correlates of plant specialization that you can see in those ant species that are symbiotic. And I'm going to briefly mention divergent estimates, divergence timing estimates I'm getting for an instance of convergent evolution within the genus. For the phylogenomic reconstruction, I am targeting reaching UCs that were designed for ants. And as the name implies, ultraconserved elements are highly conserved regions of organismal genome that are shared among distant evolutionary taxa. And when you look at the figure, we can see that for the core region of those UC loci, we don't have, those regions don't carry much phylogenetic information. But as you move away from the core towards what we call flanking regions, we have more variability in the sequence. And that's why UCs are great markers for phylogenomic reconstruction, because not only you can generate tons of them, but also you can ask based on this variability on the sequence, you can ask questions spanning from different evolutionary times. And so far, I enriched about 150 specimens of Myrmelachista, comprising 38 species. And my intent was to cover the full range of morphology and nesting habits to facilitate comparative analysis. For the ancestral state reconstruction of nesting specialization, I'm using functions from the R package APE. And for my divergence time analysis, I'm using the software called MCMC3 and two secondary calibrations and a subset of 500 loci that I obtained from my whole UC matrix. And I basically create an index of parsimony informative sites, and I capture the first 500 using functions from the R package IPS. So let's dive into the results. While I don't expect you guys to try to understand what's going on in tips, but basically this block, this gray block corresponds to Myrmelachista, and the genus was recovered as monophyletic. This is a topology obtained with the software IQ3, and most of the nodes are maximally supported. For the sake of time, I cannot really dive into the analytical part. I just want to mention that I use a partitioner method called sliding window site characteristics that use entropy-based methodology to divide the each ECU locus in 3 regions, the left flank, the right flank, and the core. And I also use the spruce up as a program that allow you to remove poorly aligned sequences. So for the rest of my talk, in order to facilitate the visualization of the tree, basically we're getting this Myrmelachista tree and I am collapsing into clades. And when I do that, we can roughly recognize 6 main clades of Myrmelachista. And right away in the top, we can see a clearly separated one that corresponds to the species found in Patagonia. So we can start to speculate about the group being originating on the region. And then you have 2 clades in brown that correspond to a smear of species moving around South and Central America. And then from that, we can have kind of like an association with the northwestern South America, and the rest of the radiation of Myrmelachista with that area of the South American continent sharing some species in different clades with Central America and the Caribbean bioregion, and that culminates with radiation of a whole clade in Central America. That's the pink block. And then with that, we can start asking questions about nesting specialization using the topology. Specifically, we can ask about how many instances of obligatory symbiosis evolved within the genus. So I want to direct you guys to the bottom left here, and then we have a color-coded legend that corresponds to if you see that or a green tree, that species is part of that first group I mentioned. That could be at either dead stem nester or live stem opportunistic species. And if you have an orange tree, that species is a live stem specialist. So when you map based on natural history data, we map that trait across the topology. You can see that a bunch of species of Myrmelachista are either dead stem nesters or live stem opportunistic species. But I also see 4 instances of live stem specialization happening within Myrmelachista. From the top, we have the first orange tree corresponds to the Devil's Garden. The second one has a question mark and corresponds to Myrmelachista cooperi. That when you see the inset, you're gonna understand why I put a question mark. I'm not so certain about that species being a specialist, because when I looked at the morphology of the queen shares several traits that we tend to associate with the parasitic life syndrome in ants. For example, the mandibles, like a pincer, and the integument is bright and shiny. And so I suspect that that species might be a temporary social parasite of another Myrmelachista species that is obligatory specialist. I still need more data to clarify that. And for other than those 2 isolated cases of potential specialization, we have everything else that is a specialist, part of the Central American clade. And that kind of like, forms a story or looks more interesting to be explored. And so for the rest of my talk, I'm gonna zoom into the Central American clade. That looks like so far we have this association of, independent evolution of life stem specialization. And so when you zoom into the clade, and what I have here is that those branches are color-coded, similar to the legend we saw in the last slide. So if the branches are green or tending to green, that species or the ancestor is either a live stem opportunistic or a dead stem nester, and the opposite, if it's tending to orange, the species or ancestor was a live stem specialist. And so basically I'm using nesting habit as a trait for this analysis of ancestor state reconstruction. And I'm sampling the character history in this case from the posterior distribution of the states, green, black, or orange, and getting a sample of this ambiguous history for the evolution of lifetime specialists within the clade. It's just reiterating that story that indeed evolution occur independently. And not only that, when we looked for the geography of those species, we see a really interesting pattern that when we look to one clade, that clade radiated in the Atlantic slope of Central America. And when we look at the other clade, that's the opposite. The clade radiated in the Pacific slope of Central America. And the story will be fascinating per se, but we can also try to find correlates of plant specialization within those specialists. And indeed, when we check the phenotypes of the queens and the workers, all the specialists shared similar phenotype in the sense of the queen searching behavior and foundation behavior, how she founds the new colony, and also the workers in the way they patrol the host plant, they clean the plant, and they protect the plant, as you can see here with the Myrmelachista cleaning up the leaf and protecting the host plant. And another thing that you can also start to think about it is before having any phylogenetic information, my parsimonious assumption was that looking for all those species in Central America that had the same behavior, occupy plants similarly, and also shared how the worker casts look. I tend to my parsimonious assumption was to associate them as part of the unique clade. And when we map those phenotypic signatures, we can indeed see that I was wrong. And I'm glad that I could merge both morphology and natural history with phylogenomic information and clarify a really fascinating story of independent evolution. And by using divergence time analysis, you can also have an idea of like the timing of those associations and when they did evolve. And when we look for the Atlantic and Pacific sides, we're basically recovering fairly the same age, and those groups are fairly young, spanning from 3 to 10 million years. So not only evolution occur independently, you have all the phenotypic signature, but also probably those instances happening concurrently. And so to wrap it up, Myrmelachista seems to be a great candidate for the study of end-plant associations, and have this fascinating instance of convergent evolution of specific relationships in Central America. And I would argue that that's possible, this scenario of evolutionary determinism on ethnic specialization within the lineage. But in order to understand those phenomena, we need basic data. So we need natural history. We need to go to the field. We also need cutting edge molecular data. But without the natural history data, I wouldn't be presenting what I just did for you guys today. So with that, I thank you for the funding sources, and I'm happy to take any questions.